Hello everyone, it's Conor here, and today we will be covering synapses. Synapses are the connecting points between two neurons. There are two types of synapses, the chemical synapse, which is the most common, and the electrical synapse. Let's cover chemical synapses to start off. Chemical synapses look like this. These are present at both neuromuscular and neuroglandular junctions, but for the sake of simplicity, I will be talking about neuron-to-neuron connection. An arriving action potential depolarizes the cell, which means it makes the membrane potential more positive, and this in turn causes voltage-gated calcium channels to open. Calcium ions then rush into the presynaptic terminal here and bind to snare proteins on what we call vesicles. These vesicles contain neurotransmitters, chemicals that act as signaling molecules. The vesicle then merges to the membrane of the presynaptic terminal and releases its neurotransmitters into the synaptic cleft. The synaptic cleft is very small, about 20 to 40 nanometers wide, which is about 1/1000th the width of a human hair. This is because neurotransmitters have to traverse this gap by diffusion, which they do in 1/1,000,000th of a second. The neurotransmitters then bind to specific receptors on the next neuron, but this isn't the only fate a neurotransmitter can have. 4 other things can happen to the neurotransmitter in the cleft. Firstly, the whole neurotransmitter protein complex can be reabsorbed into the postsynaptic terminal. Secondly, enzymes in the cleft can break down and recycle the neurotransmitters. Thirdly, specific transport proteins on the surface of glial cells can snatch the neurotransmitters, and this is currently under great scientific observation. Finally, any combinations of the aforementioned mechanisms can occur. The receptors on the next neuron that the specific neurotransmitters bind to can either be ionotropic or metabotropic. Ionotropic receptors are ligand-gated channels, meaning when the specific neurotransmitter binds to it, it opens up an ion channel which can bring us towards or away from an action potential, excitatory or inhibitory, something I will go into more depth about in the future. On the other hand, metabotropic receptors are more complicated structures. When the neurotransmitter binds, it sets off a molecular cascade or a complex chain of action, to say it simply, or even in some cases, altered gene expression. This is a topic that requires its own video, and so I will cover it in due course. Electrical synapses look like this. They are much simpler, and in turn much faster than chemical synapses, and so are found in reflex arcs. Reflex actions, such as the mammalian dive reflex, are when the electrical impulse bypasses the brain. It goes from the receptors down to the sensory neuron to a relay neuron in the spinal cord, and from there down a motor neuron to an effector, which can either be a muscle or a gland. At the electrical synapse, there is still a cleft, but there are gap junction channels which allow the action potential to just pass through. Unlike the chemical synapse, direction is not forced in an electrical synapse, and so the current can flow both ways. The main disadvantages of electrical synapses are that they are uncontrollable and get weak after many uses. Well, that's synapses. If you're still a bit confused, please make sure to rewatch the video. But other than that, thank you for watching, and I hope to see you in the next video. Bye.